Hello, everyone, and welcome to today's live broadcast, Mesenchymal Stromal or Stem Cell MSC Workflow for Efficient Clinical Translation from Automated GMP-Compliant Cell Manufacturing to Standardized Phenotyping. I'm Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Miltenyi Biotec. For more information on our sponsor, please visit www.miltenyibiotech.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Kathrin Godthardt, the Project Manager of Research and Development at Miltenyi Biotec. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Kathrin, you may now begin your presentation.
Okay, thank you very much for the introduction. I'm happy to present here today this webinar, and I would like to talk about the mesenchymal stromal cells workflow for efficient clinical translation. So this is first our disclaimer, and I continue now with some interesting facts regarding Miltenyi Biotec. So the company was founded in 1989, and up to now we have around about 3,000 employees worldwide from about 15 different nations, so it's quite an international company. I'm part of the R&D department, as these are about 23% of all the employees. So we have developed during all the years up to 70,000 different products in different research regions and also in the clinical field. So that up to now, round about 65,000 cell therapy procedures have been taken place using Miltenyi Biotec products. So about 2,500 patients were treated per year with cell products using our technology. So that shows you all that our passion is to have a nice product portfolio from benchtop to bedside. So all the products we have support researchers all over the world in the research field, but also in their way to go into the clinic later on. Come now to the part of the MSC workflow we would like to talk about today. So the improved film and xeno-free cultivation solution I would like to focus on now. So first, to come to the MSC workflow, which is worked on at many different labs all over the world. So starting with the sample, the primary tissues, so bone marrow, adipose tissue, umbilical cord, all these tissues are quite bearing some mesenchymal stromal stem cells. These cells are separated, for example, by different markers or by plastic adherents. Further, these cells are cell culture expanded in large scale or small scale. Some quality control is done, so different functions are analyzed, and later on the final cell product is maybe cryopreserved or directly used for the different applications. So what Miltenyi is doing is that we are providing a lot of different products to support these MSC workflow, and even here it's quite important for us to have this seamless translation from basic research to clinical research. And to show you in more details how this look like, I would like to focus on the MSC Isolation and Expansion. So always questions come up. So what is needed to overcome challenges when working in MSC field? And the main problems here are the improvement of cell culture products, to have better methods for expansion of MSCs, and to get some help in complying with the relevant GMP guidelines or regulations. So we are focusing here to support all these needs, and therefore we have developed different cell culture media for MSC Isolation and Expansion. For example, we have these FCS-containing media and also xeno-free media, so xeno and serum-free media. And I would like to point out that we have one medium in a GMP grade to support also all the higher regulated needs. And to show here one very important feature. So if the customer or if researchers start in the research field, they can use the xeno-free StemMACS MSC expansion media XF, which is xeno and serum free. No additives are necessary here for adhesion of MSCs to the plastic, so no coating of surfaces. And we have the same formulation are also available as the GMP version of this media. So even this media is also xeno and serum and also phenol red free. It's supporting an efficient expansion of human MSCs from different tissue sources, also from the primary tissue. No substrate pre-coating of the culture vessels is required. And as you can see here, we have flexible bag sizes available. So this is the 2-liter bag, which you can see on the picture with the corresponding supplements. And also a 500 milliliter bag is provided. This medium was developed in compliance with relevant GMP guidelines, designed following the recommendations of USP 1043 on ancillary materials, and it's manufactured and tested under an ISO 13485 compliant quality management system. So I'd like to show now some more details about our quality analysis of this media. So we have done a lot of experiments here to show that MSCs are working quite nice after expansion. So first here we wanted to show how the clonogenic potential looks like of our StemMACS MSC XF medium, as well as the GMP version of this medium, the MSC-Brew GMP medium, in comparison to the homebrew alpha MEM and DMEM media, as well as a comparison to 2 commercially available cell culture media. So as you can see here, mainly commercially available media need some input of AB serum to support the adherence of MSCs coming from primary tissue. This is not the case for our StemMACS XF and the MSC-Brew medium, so we have better MSC adhesion, results in a higher number of CFUF colonies using the StemMACS medium and the MSC-Brew. And furthermore, there is better MSC proliferation, resulting in larger colonies. So beside the number of colonies, we have analyzed here the number of MSCs within one dish. This is depicted within the dots in this graphic, and yes, as you can see here, the Miltenyi media are supporting the proliferation even at the beginning of the culture. Quite nice. Also, if you are interested in a clinical scale expansion of MSCs, so to say about generating 200 million MSCs, it's possible to do this quite fast. So just within 15 days, starting with 10 milliliter of human bone marrow, using the StemMACS medium and the MSC-Brew, you will reach this high number of cells compared to the homebrew DMEM and alpha-MEM media, which takes a lot more, so more than 3 weeks roundabout to get this high number of cells. So the positive effect here that we achieve a clinical scale MSC number with less media consumption. So we are just using 50 to 70% of the media which is normally used for homebrew media. And the reason for that is because we are saving about 14 days, 15 days after reaching this high number of MSCs with the MSC-Brew GMP media. But not only this a low passaging cultivation process is optimal. We also looked for large-scale expansion, so also for higher passages. So MSC-Brew GMP medium allows for stable expansion of MSCs. Here we have looked for this for up to 6 passages, and the proliferation range is quite nice, quite comparable with both Miltenyi media, and it's improved compared to the homebrew alpha-MEM and DMEM media. But what's going on with the cells then after expansion? So of course this is a question, how functional are the cells? So therefore we have done some functional assays. One, of course, very important topic is the expression of MSC-specific surface markers. For this purpose, we have used the MSC Phenotyping Kit, a flow cocktail, including different markers to stain cell surface and antigens analyzed then by flow cytometry, and we can show here that the relevant MSC markers are expressed by the cells. Even also the differentiation media were used for differentiation capability, so for adipocyte differentiation, osteoblast differentiation, and chondrocyte differentiation. What is also quite interesting for many researchers is the immunomodulatory potential of MSCs after expansion. So here we use the MSC suppression inspector to look for the T-cell suppressive potential of MSCs. And as you can see here, the potential is quite preserved and quite high compared to the homebrew media shown here in this graphic. What is new here is we have updated the MSC Phenotyping Kit, and here now I would like to show you in more detail how standardized and reliable this phenotyping cocktail is. So the design of the MSC Phenotyping Kit is as follows. So you can do around about 50 tests with one kit. The kit is consisting of an MSC phenotyping cocktail and an isotype cocktail, as well as 6 individual antibodies for compensation control. So this is quite important to get a reliable result at the end. The phenotyping cocktail contains all the relevant MSC markers, CD73, CD90, CD105, as well as the non-MSC markers and the HLA-DR, as recommended by the ICT consortium. So it facilitates standardized and reliable phenotyping of cultured human MSCs from different tissue sources. This is just one example showing human MSCs cultured in StemMACS MSC-XF medium and analyzed by the MSC Phenotyping Kit, showing a high expression of CD73, 90, and 105. The non-MSC markers are negative, and which is also a nice feature that the HLA-DR antibody, which is in most cases also negatively expressed on the cells, can be analyzed with a separate color here. And this is quite important because sometimes MSCs are stimulated, like you can see here. So the stimulation can be done by interferon gamma or TNF-alpha to have a look of the MSC function in more detail, but sometimes it's also the case when, for example, in FCS containing media, a high amount of interferon gamma is present, then sometimes the HLA-DR signal is quite high. So therefore it's really nice to have this marker with a separate color here in this kit to analyze it separately from the other non-MSC markers. Because as you see here, before stimulation it's negative, and after stimulation with interferon gamma and TNF-alpha, the signal is quite high. The hybridoma antibodies are quite an old technology. They were used in the old antibody kits. So this technology is from 1970. Antibodies are produced as a hybridoma in mouse and then later on in the hybridoma cell culture. So the new way to produce antibodies in a recombinant way, using here our reaffinity antibodies, is based on a system of a mammalian cell production system producing recombinant antibodies, antibodies consisting partly of a human and a mouse part. And there you have some benefits of these reaffinity antibodies for cell analysis. So first, they are recombinant generated, and they have one universal IgG1 isotype that improves, of course, the reproducibility of the assays in the lab later on. Furthermore, there is a mutated FC region, and this improves or reduces the background signal. So summing up these, the kit is quite easy to use. The 5-chiral antibody cocktail includes 3 positive and 5 negative antibodies for MSC phenotyping. In addition, an isotype control cocktail and 6 individual antibodies for compensation control are also included. The assay is very standardized because antibodies are included that are defined by the ISCT consortium for MSC characterization by flow cytometry. There's a really high lot-to-lot consistency and reproducibility because all the used antibodies are reaffinity recombinant antibodies. The kit is very reliable because we use different flow chromes for the HLA-DR antibody to distinguish between activated and non-activated MSCs. I would like to come up now to the next topic, which is sorting MSCs via the MACSQuant Tyto. So sorting cells bear several problems. So for example, when you sort cells with a cell sorter, there's high pressure and shear stress affecting the viability and function of cells. Decontamination, cleaning of fluidics required after each run. there is a high chance of carryover between different runs, and even when we are talking here about these rare cells, carryover is quite an important topic. Adjustment of drop delay and laser alignment is needed. So the decompression of cells affects the viability and the functionality. Application of charge affects viability and function and might alter gene expression. Safety concerns due to aerosol forming or alternatively working under huge biosafety cabinets. Contamination of the sample affects downstream processing. So all these points are quite problematic, and therefore we have now the MACSQuant Tyto. It's with its unique features. So it's really gentle to the cells because it has very low pressure, no decompression, enables resorting of functional cells, and we have no cell loss intubing sets because the whole procedure takes place in the cartridge. And all cells are remaining in this cartridge. The MACSQuant Tyto is fully sterile, so cartridge provides a closed system. So the cartridge system also gives some opportunity not to have any carryover from experiment to experiment because you need and use a separate cartridge for each experiment. And we do not have any contamination of fluidics. So it's fully safe system, the absence of aerosol and droplets, and no biosafety cabinet is needed. The device is really easy to use, so it's a benchtop device, no adjustment of drop, delay, or laser alignment needed, and no cleaning required. So this cell sorting process here at the Tyto is shown in more detail in this picture. So you really have, no pressure, no charge, less aerosol or no aerosol development. So at the end, cells are quite feeling well, are very viable and not activated in any kind of process within here. So for the MSCs, we have done several experiments to use this device, the MACSQuant Tyto, for sorting using CD271 and anti-MSCA-1 as specific markers expressed on MSCs coming from bone marrow. So we used the bone marrow mononuclear cell fraction labeled with CD271 PE and anti-MSCA-1 APC, and you can see here before separation this is a really low frequent cell population with only 0.04%, and after separation there's a really high enrichment of cells of more than 90%. Also the viability is quite nice, and if you then put these cells after sorting and also compared to the situation before sorting in culture, you can see that there's a high increase in clonogenic potential in these sorted cell population. Here we analyzed the ability of cells to expand and proliferate after the sorting process. We have passaged the cells several times, and we can show here that the sorted cells grow in a comparable manner like the plastic adherent cells. Also, the marker expression is as it should be for MSCs, and also here we looked for the T cell suppression potential. So it's quite preserved and comparable before and after sorting. But besides sorting, of course, the cultivation process is still important, and many people would like to do here a closed procedure, an automated procedure. So therefore, we are providing the new CliniMACS Prodigy adherent cell culture system. So GMP-compliant cell manufacturing is really complex workflow, including a lot of many different processes. So cell preparation, surface coating, cell cultivation, media change, harvesting, and for all these processes, many different devices are needed, and as well, everything has to be done in clean rooms. So we would like combine all these steps within one machine within the CliniMACS Prodigy to have the integration of these various cell processing steps. So the hardware here, some description on that. The CliniMACS Prodigy platform is an integrated and closed system for automated cell processing from starting material to final cellular product. What we can do is sample preparation, for example, the density gradient centrifugation to isolate MSCs from bone marrow, for example. Cell washing is possible, media change, magnetic cell separation, cell stimulation, genetic modification, and final product formulation. So the closed system from sample to product, that is what we want. So therefore we have the single-use disposable tubing set, sterile filters at input ports. There is possibility of sterile welding, for example using the Terumo device. Integrated QC sample pouches are integrated within the tubing set to make it easier for QC sampling and doing assays in between, taking in-process controls during the expansion process. So for cultivation, we have several options. First, the Centricult Unit. You can see it here on the right side. The surface area is around 100 square centimeter for adherent and non-adherent cell cultivation. Automated feeding and harvesting processes are possible here. So we have a temperature control because the Centricult Unit is placed within the device and is part of the whole tubing set. And the device is also supporting this Centricult Unit with a defined atmosphere, so CO2, for example, 5% CO2, 10% CO2, whatever the cultivation requires. There's visual cell examination possible by an integrated microscope. The microscope is also part of the CliniMACS Prodigy. And we have multiple input ports for different media or cytokines. Of course, these 100 square centimeters might not to be sufficient for all the different applications, especially for the MSC workflow. It is not sufficient. So therefore here, customers have the possibility to connect an external culture vessel to the tubing set. And then it's possible to have a scalable cell manufacturing process. Now coming to the tubing set, it's GMP compliant, and it's possible to do sterile welding during the process. It has integrated several bags and sample pouches for passaging and QC. It's easy to connect an external cultivation vessel, as it is shown here, via a dead end tube. And you can not only connect one, for example, cell stack to these dead end, you can also connect up to 3 cell stacks, 5-layer cell stacks, for example. The whole tubing set is compatible with the upcoming CliniMACS electroporator for gene manipulation. So showing now here in more detail the GMP-compliant MSC expansion workflow using the CliniMACS Prodigy adherent cell culture system. So everything starts again also here with the bone marrow. and the whole process then is automated within the machine. So having the density gradient, centrifugation, seeding of cells, immediate change, harvesting, and seeding again in different cell culture, external cell culture vessels, up to the last harvest of the target cells. So the process is quite handy and quite optimized to reduce the manual hands-on time. So here within this process, we have reduced the hands-on time to about 70%. The process is not only available for bone marrow, to isolate MSCs from bone marrow, but also you can do this with enzymatic digested umbilical cord or enzymatic digested adipose tissue, and also here you of course find the same reduced hands-on time because of all the automation of the different steps. So we prepared again here some data on using this Prodigy process for manufacturing of MSCs in large scale from bone marrow, umbilical cord, and adipose tissue. And in all cases, you can see quite nice that with a low amount of primary starting material, after not more than 2 or 3 passages, we end up in a quite high amount of MSCs. Up to 700 million MSCs were prepared here. But of course, this number can extend it, of course, on the one hand by starting with a higher amount of starting volume or extending the cultivation period. So here in these experiments, we cultivated the cells for up to 14 or 15, 16 days. So summing up now, we have a complete workflow for efficient clinical translation. The workflow is sterile and gentle, for example, for cell sorting of MSCs using the MACSQuant Tyto. We have an efficient MSC expansion with serum-free and xeno-free MSC-Brew GMP Medium. And we have talked about the GMP-compliant MSC expansion using the CliniMACS Prodigy Adherent cell culture system. Using the MSC Phenotyping Kit, it's possible to do it standardized and reliable phenotyping and analysis of MSCs after cultivation. So you see here now again the MSC workflow and all the different solutions we have talked about during this webinar to support the MSC workflow, for example, for preparation of the primary tissue using the GentleMACS, for example, for umbilical cord dissociation, for doing the cell preparation using the CliniMACS Prodigy or the MACSQuant Tyto, cell expansion using the GMP-compliant cell culture media or different cytokines, doing the quality control using the MSC Phenotyping Kit based on the flow analysis, doing some cryopreservation of cells afterwards, and then having the final product available. So I would like to thank you now for attending this workshop. Thank you very much.
Thank you, Kathrin, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. All right, let's go ahead and get started. Our first question is, compared to other commercially available kits, what are the advantages of your MSC Phenotyping Kit?
Okay, so our MSC Phenotyping Kit includes 3 positive markers and a set of negative markers, including CD14, CD20, CD34, and CD45. The cocktail does contain PE, fits APC, and it also includes a separate negative marker, HLA-DR, which is visible with another color beside the other negative marker. And this makes it possible to analyze cells which are stimulated or not stimulated via the HLA-DR expression of cells. Furthermore, we have included here quite high standardized recombinant reaffinity antibodies, which improve the reliability and the specificity of this antibody cocktail.
Great, thank you. Okay, next question says, do you have other kits or antibodies to test the function of cultured MSCs?
So yes, we have. So beside the phenotyping or the marker expression, a lot of people in the field are interested in analyzing the immune modulatory potential of MSCs. And here we are providing the MSC suppression inspector. This is a kit to look for the potential of MSCs to suppress T-cell proliferation, and this is quite a very important function of MSCs and necessary for a lot of different clinical applications where MSCs are currently investigated in. Furthermore, beside the suppression inspector, we are providing differentiation media for adipocyte differentiation, osteoblast differentiation, and chondrocyte differentiation.
Great, thank you. Now, in your opinion, for which application sorting approach is helpful, and can you give us some examples?
Okay, yeah, I think so. The sorting of MSCs is very important in all cases where you would like to analyze the primary MSCs in more detail. So the primary MSCs which are in the primary tissue, in the bone marrow, in the adipose tissue, or in the umbilical cord, for example. Because here in this tissue, the MSC number is really, really, really low. So as we have seen in the presentation before, the frequency is around about 0.01 to 0.05% roundabout. And to analyze these cells in more detail, a sorting approach is quite helpful. For example, if you would like to do gene array analysis or something comparable with these primary MSCs. So only with this sorting approach you get a high purity of these uncultivated, untouched cells. And there are different markers which can be used for this approach. For example, CD271 and the MSCA-1. Of course, another reason why people would like to sort MSCs is, of course, if they have defined a specific antibody for a subpopulation of MSCs, primary MSCs or cultured MSCs. This could be another interesting example for a sorting approach.
Thank you. Okay, looks like we have time for one more question. And we will finish with this question. Can you talk a little bit more about cell yield using your platform to culture MSCs? And to what extent can you upscale the system?
Okay. Sorry, can we repeat this question? Yeah, please repeat this question.
Sure. Can you talk a bit more about cell yield using your platform to culture MSCs? And the second part of the question says, to what extent can you upscale the system?
Okay, yeah, that's also a good question. So in our experiments, we have cultured the MSCs from the different tissues to up to 700 million cells. And for this approach, we have used 2 or 3 5-layer stacks, depending on the starting material. But of course, it could be upscaled even more. So if you would like to start with a higher amount of cells at the beginning or to continue the cultivation period, even then you can extend the whole system by using 6 5-layer stacks. And these would then result in a surface area of about 20,000 square centimeters.
Well, then I'd like to thank the audience for joining us today and for their questions. Any questions we did not have time for today and those submitting during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to once again thank Kathrin for her time today and her important research. We would also like to thank LabRoots and our sponsor Miltenyi Biotec for underwriting today's educational webcast. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.